Welcome back to the Soil Matters, guys. We have a very special guest, Dr. Elaine Ingham, and somebody told us something very special about you, Elaine. So, boys, are your voices ready?
Let's go.
1, 2, 3. Happy birthday to you. Happy birthday to you. Happy birthday. Happy birthday to you.
And many more.
I think we should get our day jobs.
Even closest.
Or at least
Everybody
we're gonna.
knows who Dr. Elaine is.
Oh goodness. Uh-oh.
No, that's what Ken does. He's the man behind the curtain, so he pops on and off.
The Wizard of Oz, they call me.
Aha.
I believe Ken does call himself that.
So I don't know where to begin. Obviously, you need zero introduction, Elaine. So welcome to the show, and why don't you kind of bring us up to speed on all of the work that you're doing?
Oh goodness. It's one of those cases of we thought we would have to write 5 or 6 proposals in order to get 1 or 2 maybe funded. Yeah, we've got all 5 or 6 funded. So now we're looking for people to come and help us do all the work that we're going to be doing, and so that's pretty good. We're doing a lot of work. Of course, the school, Soil Food Web School, and Soil Food Web Inc. are working very closely together, but they are indeed separate entities. So the school does what you would expect. It teaches people how to understand what's going on in their soils and what to do if they don't have enough of the microorganisms needed to perform the jobs. So the Soil Food Web Incorporated is for doing the research. We have barely begun to understand what's going on in the soil. We've got some pretty good ideas on some very important parts of what goes on in the soil with all these organisms and how it affects plants, but we're a long way from knowing what everything means. So keeping us busy. I'm going to have the research being done at Soil Food Web Inc. and the school and the certifications, all of that at the Soil Food Web School. I do like to point out the website that we have that gives you a little bit of understanding of the classes themselves, where we have the foundation courses where we teach everybody about the various things we do know about interacting the biology with the soil and the organisms and the plants. So all those real important concepts that you have to understand the specifics. So then Foundation Class 2 is going to be the teaching you about how to make real compost as compared to the waste material that people have called compost for a long time. There's every aspect of making compost has to be observed, and you have to do it right. You've got to have the recipe correct, or you won't get the heating, you won't get the killing required of the disease and pest and problem organisms. We want all the beneficials. We don't want the not beneficials, things that are going to kill our plants. And so we teach people how to make compost, and then we may teach them how to make extracts and teas from that, so that it's a lot easier to apply soluble nutrients or in solution, a lot easier to add things in water, unless you're right at the beginning of the whole food web. And so we've got some fun things going on the picture on what you're seeing on the board, I assume. So kind of catching up. We can go through all of this if you want, but we might not want to do that. then after the compost and teas, we teach people how to use the microscope. And you have to have the ability to determine whether your plants are going to grow as rapidly as they can, whether they're going to be resistant to the diseases and pests and problem organisms. You've got to know if the root systems are going down deep, and the only way to make certain of that is to know that you've got the right biology in the soil. A lot of people will um, uh, gosh, I lost my. Can you tell that I'm 71 years old today?
Not at all. I thought you were 30 at least,
Oh, he is, giving me lots of leeway, huh?
It happens all the time, Elaine. It's because, when you start talking about these really complex and deep things, it's very easy to get sidetracked.
And
So no
your
stress whatsoever.
your brain goes off, "Oh, I've got to tell him about this." Well, just wait a minute until I finish this.
Exactly.
Yeah, and it's like, "Stop it, brain, stop."
So maybe this will help. Are you in your new farm?
Not yet. Because of the last craziness that occurred with federal funds. Looks like we were not going to be able to pay anything off. That the people that were going to be giving money for the compost or for the place, the farm that we were going to move into, they're being a little more calm and cool and collected, and they want to know what all is going on. So now they're back to, okay, we'll go ahead and pay for the Ramsey's farm and actually for the Tainable is the name of the company that he has for helping people get going with important concepts and teaching folks hands-on is something else that we do at the Soil Food Web Inc. So I think that kind of brings me up to date, more or less. Lots of things happening overseas.
Yeah. Why don't you tell us a little bit about what you're doing overseas and of course in Sri Lanka, one of the most incredibly beautiful and diverse places on the entire planet.
Yep. So we're working with growers down there to teach them how to use the proper biology, how to make compost using the principles that we were talking about, learning how to look at the soil and determine whether they've got all of the organisms present in the soil that the plant is going to need to stay healthy. So what is a healthy plant? It is really defined by the sets of microorganisms that are present in the soil and how those interactions with the plants, with the bacteria and the fungi and the protozoa and the nematodes, the microarthropods and the encotraids and all those good guys in the soil. So we teach people how to identify those down to functional level. We've found that you don't really need to know whether you have Pseudomonas passomobilis or you have Pseudomonas aeruginoso or any one of the other. Well, gosh, there's how many different species of bacteria on this planet? It's mind-boggling. And so which are the ones that are supposed to be in your soil? We don't have to know every single one. We just have to make certain that they're all beneficial. And so we control the conditions in the soil to make certain that all of the organisms growing in that mix of food resources for the bacteria and the fungi, the protozoa, of course, the nematodes, bacterial feeding nematodes are eating the bacteria, amazingly enough. The fungal feeding organisms are preyed upon by fungal feeding nematodes, fungal feeding microarthropods. So the whole list of organisms in the soil. So we've been working on that. Of course, we've got the school now, and in the school, we're teaching students. We've had, I think it's something like 200 students have gone through to the consultant stage so that they can help you. Where do you go to find these organisms? These organisms, people? Where do you go to find these people? You go to our website, soilfoodweb.com, and you want to look for the list of people who have passed all of the classes. They've done very well. They've had to do a whole sequence of um, building up the soil biology, getting it to the right balances for the plant so it grows to its maximum capacity. So there we go. There's, you can see part of the world there, but you can see there was a couple of people in India. If you get down into Sri Lanka, there's a couple people in there. Through New Mexico, through South America, you can, yeah. So here's all the people that have been learning all of these things and can help. This is this looks like a little bit of an older picture. So, or I haven't been looking all the time to see who's actually listed. So we've got a number of people that can help around the world. Doesn't really matter where you are. You can take these. Um, you can take these fungal
The network. Yes, you can use the network to find people close to you. As a matter of fact, I thank you for that. I mean, I do all my own qualitative, but sometimes I need quantitatives. And so I started working with one of your graduates from, I think, a couple years ago now, Andy Marsh. And she's just been an absolute pleasure to work with. Her reports are just so thoughtful. She gives you ideas, explains what these ratios actually mean. So it's been an absolute pleasure. I mean, you're doing a wonderful job educating these people in a way that provides us, the end users, some really, really good data to work from.
Yep. And that's what we're trying to do, is achieve those goals so everybody can have a mentor, have somebody helping them whenever they need help. So
Yeah,
thank
and
you, Leighton, for
yeah, and you know what's great, Elaine, is the successions. Like when you start working with the soil food web consultant, you can track your year-to-year growth, which is something that we really didn't have a lot of tools to do unless you went through that whole course, which wasn't really even available that long ago. I mean, it's only been what, 3 or 4 years since you really got that thing off the ground.
Yeah, we have really developed a really good group of people that work together well and really caught on to the whole reason why we were looking at all of these organisms in the soil, because it tells you what your plant health is. It tells you if your plants are going to be able to produce a much higher productivity than they could do with the inorganic pesticides, inorganic fertilizers, and all of those things. And we routinely have an increase of at least 20 to 50% increase in productivity. Quite a bit of the time we will have increased productivity up to 100 to 300%. And then, of course, there are those real outstanding people working with the organisms in their soils and getting increase in productivity of up to 1,000%.
I know when you use these numbers, people don't believe you.
Yeah,
But
they just
I'm a witness. I've seen it time and time and time again, based on the foundation that you gave me. You remember that old stainless steel kettle I called Raquel?
no, I don't. I'm
Well,
sorry.
remember I had this Sandy the stripper up at in April's greenhouse, remember? And then I moved on to Raquel, you know, obviously another stripper. Well, I recently located that to a farm in the Midwest, and the gentleman's treating 500 acres of corn and beans. And guess where his corn is right now, Elaine? It's supposed to be knee high by the 4th of July. Well, guess where his is? Waist high. And it's
Waist high.
literally waist high.
Mhm.
And all around him are droughts.
Yep.
Like he's in the drought, but his plants are growing where all the nitrogen plants are curling up and dying. And so, you know, this is just like another thing that blows his mind because he's relatively new to it. He did 200 acres last year. And listen to this, Elaine, you're going to love this. He only did one application of fish brew. He didn't even use Raquel because he didn't have it yet. So it wasn't a worm castings, biocomplete compost, and fish brew. So his cover crop wasn't tall enough that he could roll or crimp it. So he called me in a panic and he said, "Look, I got to till it." And I'm like, "Well, just keep your tillers, you know, only like 1 inch so you don't penetrate into the soil too deep." Because you have all of these wonderful beneficials there. And I understand that if you don't do something, then it's going to outcompete your cash crop. So he tilled it 1 inch, and his neighbor sees him out there walking around looking at the clods. And he's like picking up clods, and he's like, "My soil usually runs between my fingers. Why is it clumpy?" Right? and then it gets worse or better. He's like looking at the clods, and it's loaded with worms. So his neighbor comes over and he's like, what are you doing? You're looking like a crazy man out here. And he goes, "Pick up one of those clods. It's full of worms." And this guy's a 74-year-old farmer.
Mm-hmm.
Been on that farm since he was a little boy, and he's never seen an earthworm. 1 year, Elaine. 1 year.
1 year, yeah. And that's usually what it takes, as long as you realize that it's the biology that you're trying to improve. And so you want to know what is good for your plant. What's the bacteria minimum amount of bacteria that you need to have? You want to have a minimum amount of beneficial fungi. You want the right sets of protozoa, nematodes, and you don't really want to see a whole lot of ciliates, because that's a little message from Mother Nature coming to you that things are getting a little bit anaerobic in your soil. And maybe you want to start looking at what you need to do to get that rebuilt. The aggregate structure in that soil is not holding up. So how are you going to get it to hold up? And that means putting on another application of a compost extract. That can go through your irrigation system. You can apply it using drones. You can apply it with a sprayer, whatever you want. So, you know, pick the equipment you already have and make sure the insides are cleaned out really well, because you don't want any toxics left over, killing your organisms. You want them to all be alive, and then you'll see a reversal of the ciliates being present. And so, of course, all of these organisms need to be there. Keep saying that over and over again, because how do you know if they're there? You have to use your microscope. But it's not that difficult a process. People try to make it really difficult, and you have to identify everything to the genus, species. No, you don't. Don't kill yourself. That's just nonsense. We want to know what the function is. And so we'll be watching for the shifts in the bacteria, in the fungi, the protozoa, the nematodes. And you'll see that as those shifts start lining up with the stage of succession that your plants, your crop needs, then you'll really see an improvement in yields.
I'm into that. Now, you hit on something that's really important, and I can't stress this enough, about cleaning out the equipment, right? So many people make that mistake of using old farm equipment, and then they just start running compost teas through, expecting these miracles, when in reality, they just killed everything that they put in there. Mm-hmm. So what do you recommend for these farmers who are going to transition into the biological world for cleaning out all of those pipes and tanks and pumps? I mean, is there a recipe that you could share with us?
It's typically hydrogen peroxide, and you want about a 3% solution of hydrogen peroxide. So you just go to, you know, your local drugstore and get a container of, you know, 10% hydrogen peroxide. You can dilute it a little bit down to 3%, and then just spray, you know, the sides of the roads, or, you know, where you don't particularly care. Just use that material. to clean and wash, or maybe the hydrogen peroxide, because the hydrogen peroxide reacts with just about anything and has that cleaning solution effect where it's pulling all of that yucky stuff off of your off of the inside surface, inside the pipes, inside the sprayer units, and you're left with clean machinery. So we do try to get people to not buy things that have a 90%, 95% bend in it, because as the liquid with your compost tea, your compost extract organisms in it, it's coming along and it hits that turn in the pipe, and it all these organisms in there smash into that upper part of the pipe, and then all the dead bodies end up over here, and you've lost perhaps a significant amount of your required organisms, and you don't see what what was going wrong. Now, if you're looking and checking with everything with the microscope, you just have to take a pipette of that liquid, put it in a test tube, line up the test tubes from all the places that you wanted to see if you got it clean enough, and you know, shake a little before you take it out and put a drop on your microscope slide, and you look at it and you go, there's no biology in here at all. Oh, oh, I
Problem.
yeah, big problem, because now all that other work that you did is not going to be able to support the problem. You're going to end up with things you don't want happening. What you want is look at that very first slide and go, whoa, look at all that. It's just zooming all over the place. It's I can't count however many I've got in there. I can't count the number of bacteria, protozoa, nematodes. Okay, that's great. You've got more than enough. And that's all you really have to record is it's more than enough. So then you look at your second sample and you'll notice, you know, it's improved or it's gotten lower, and you have to understand where you are in that successional process. And that's why we always want to have mentors working with our students. We always want consultants. Once you finish up all of the classes and you're ready to go out and apply all this, you might want to find a close enough consultant to help remind you of what of the things that you forgot about. How could that happen? Well, yeah, I can talk to you about 75.
Well, I'm only pushing 60 and I'm gone.
Yep, as long as it's not 71 per day or per hour. Uh, that one of the joys of getting older.
Oh, yeah, tell me about it. So, tell us what you're doing in Sri Lanka. So you're teaching farmers how to use your methodology for crop production or forest restoration or everything? Because I
For
know
in Sri Lanka, it's focused very strongly on food production because
okay.
they had such a horrific experience. It's got to be just a little bit over a year or so ago, where they basically were up a creek without a handle. We came in and we started showing people what to do, and there are, you know, definitely people who are utilizing that knowledge already. So we're working with a lot of different growers and a lot of different um, with
Plants,
a lot of different plants. Yeah.
crops, yeah.
Yep.
Interesting.
And same thing, you know, it's going to happen in other parts of the world. So, New Zealand, Australia, India, South Africa, Europe, we have a lot of people really climbing on board there. And so, you know, look up the folks who are doing the work in that part of the world. It's amazing. When we have our next webinar, um, uh, if you're signed up, I believe you can, if you're signed up for the classes, I believe you'll be able to watch this. And it's just incredible what happened with the grapevines. So just beyond anything they could believe, anything they could hope for. We have some projects going on with reforestation in a number of places around the world, in the United States, as well as in Central and South America, as well as in the Southeast Asia. So just too many things to talk about. I need to, clone myself.
Yes, you do. And hurry up. Need about 20 of you. So what did you find in grapevines? Because that's always been such a big issue. The way they treat these vineyards is deplorable, to say the least. But
well, yeah, it's the problem is that they're out there putting on inorganic fertilizers to begin with. That kills 50% of the organisms that need to be in the soil. If you had those organisms, kept them alive, you wouldn't have the problems that you do have. Well, if you till, you're going to be destroying 50% of the organisms in your soil again. And so once again, you've kind of defeated yourself, because which does more bad things to happen, which causes what kinds of percentages are we talking about when we're dealing with something that is beneficial? So we'll be improving the biology in the soil every time we put another application of that compost or extract or tea down. But we're only going to recover maybe 45 to 50%. When we're dealing with any of the toxic chemicals, they're all salts, and they will take water away from your microorganisms. They take water away from your plants. So don't we understand this enough already? But people keep doing this. So, how much do you lose? So 50% or more of your beneficials are going to be killed outright whenever you use those toxic chemicals. So now you're going to have to start putting that all back again. Don't think that you really need to do that. Just decide to go to the biologically complete condition and get all of these things up and going. All you really need is some good compost. If you don't have any good compost, then I'd say don't even start, because you've got to have those organisms ready to go into the soil and be utilized by those organisms to help them grow and thrive, to help your plant grow and thrive, and determine that you've got enough organisms to be able to resist diseases and the pests. And we show over and over and over again that you just have to get the biology right, and all of these bad creatures go away. You cannot let anything go anaerobic. You have to know what kind of foods are required by different kinds of organisms. What high temperatures mean. All the, we need to write a book that says, here's Mother Nature, here's what Mother Nature is trying to tell you when you see ... and just go through it, A through Z, or A all the way through the end of the alphabet, what it means to be dealing. How are you going to deal with these organisms, this interaction, and make certain that the good guys win?
Yep. So biomimicry, right? Like, what does nature do? She doesn't water her plants. She doesn't fertilize her plants. They take care of each other because they grew up together. And that's unfortunately what we've done with monocultures caused the problems that we deal with in this day and age.
Yeah. You've made sure that you're not going to hold on to any water.
Yeah.
It's just going to go erosion. Your nutrients, of course, are soluble when you put those inorganic fertilizers in. And so all of those inorganic fertilizers are just washing down the hill, and they're going to cause problems now for everybody in the rivers and lakes and streams and the oceans and the seas. We're destroying ourselves.
Literally.
We like to pay attention to what Mother Nature is telling us. And just like you said, she's got a whole bunch of tricks to make you pay attention. But if you ignore her, she comes up with a nastier
Oh yeah.
Just like a 2-year-old, if you take my toy from me, I will hit you twice. If you take it away from me, I will hit you 3 times.
Yeah,
Continue.
I remember some of the work that we did back at Rodale and like studying soil temperatures and stuff. And people just don't get it. If there's a cover crop, that soil stays like really nice and ambient, 70, 72, 68 degrees. But as soon as you expose it to sunlight, man, it heats up and cooks and kills all of the biology that you just put down. So, these little tricks like that, and polyculture, regenerative agriculture, et cetera, is the real way to work with Mother Nature, not try to fight her. But
Yep. Because who's gonna win? Who's gonna win that argument?
every time she is, every time,
Yeah.
no questions asked. Av, I'm sorry, I've been hogging the microphone. You want to jump in here, like?
Well, it's been 20 years since I took the course with.
We've learned a few things of.
I guess that's where I'd like to lead to. Can you summarize the last 20 years? No, I'm just wondering. We're starting to. I mean, even with this show, we talk about KNF and we talk about biodynamic. We're talking about Jadam. Leighton has talked a lot about Olivier Hassan's work and looking at electric potential. Redox. We've had Jim White come and talk about the rhizophagy cycle. I attribute a lot of this to you, Elaine. You definitely were the spearheaded bringing biology into the equation, and it's because of your pioneering work that we're starting to really realize that you don't need 150 pounds of nitrogen per acre because actually the microbes are going to make you 175. Do you want to just comment on any aspect of some of your major learnings as you're seeing people that you've inspired do some more work around biology and what you're learning from them as well?
Yeah, it's too big a time in my life to try to go through and really pick out all of the things. But when you go back to the original classes that we started to do back when I was at Oregon State University, we had 5 really big things that we could do to help fix the biology in the soil. But we just talked about those 5 things because that's all we really had data for at that time. Well, with constantly looking at what the biology is doing in the soil, trying to pull out, what does the curve look like? It starts off way over here and it's slowly like, that's a good sign usually, unless that's a disease causer that you were assessing. But we started to understand more about how everything fits together and how it works. So, like with the classic work that I did, coming from the years at Colorado State University and at the University of Georgia, we knew that we had to have these predator-prey interactions. The plant actually gives off exudates and tells the bacteria and fungi, or certain of the bacteria and fungi, of the things that the plant requires. And so the bacteria and the fungi go, okay, I can do that. As long as you give me energy right here at the very beginning, then they will do that work of going to the surface of a rock or a pebble, gravel, boulder, parent material, whatever you want to talk about. And they will use their enzymes to pull those nutrients that the plant asked for. And they store it inside the cells of those bacteria and those fungi. So they are now in an organic form. They are soluble. Organisms can take them up. So then when the predators of the bacteria and fungi come along looking for dinner, they'll chow down on the bacteria and fungi that had that elevated amount of nutrient for the plant. And so when that's released in a plant available form, the plant can take it up without expending any energy at all for getting that inside of the root system. So most of the nutrient cycling, we pretty well had a well in hand. 65 to 75% of the nutrients that your plant requires are going to be given to that plant through this process that I just told you about. And that's 65 to 75% of the nutrients that your plant requires. That's a pretty huge improvement.
Very significant. Yes.
Yeah. So, but then we really needed to get the last 25%. So what else does this nutrient cycling? Well, it was pretty obvious for in a few places, mycorrhizal fungi. They're taking up the nutrients that the plant requires. And again, here's the plant telling the mycorrhizal fungi what it needs. And the plant now says, I ain't giving you no energy, boy, until you bring back the nutrient that I want. So see, the plant's learned a little bit. And there's less of the organisms are in charge, the plant's in charge. And so here come those nutrients back. The nutrients are traded for energy once within that intersection of the um, cell walls and the cell membrane. And so the plant gets the nutrients that it wants. The fungus gets the energy that it needs. And then the plant says, well, now don't be so hasty. Stay here and I'll tell you what I need next. And they work out the deal. Out goes the fungus. The plant pulls in the energy and the sunlight energy in the form of uh, uh, a material that that the bacteria or fungi could eat. So that filled in a little bit more of our understanding of how plants get their nutrients. But we were still sitting there with like 5 to 10% where's it coming from, guys? It's probably coming in exactly how, where's it coming? And that's where James White's work with rhizophagy was like, yes, we've got it all figured out. We know what we need to have in that soil, and we know how much of these different organisms that we need in order to reach maximum productive capacity. So, rhizophagy is basically where the root coming down into the point at the end attracts the bacteria and fungi to move into that area just below the root and take up these little tidbits, kind of like the plant was putting in the the understanding what it needed and luring in the bacteria and fungi. And when the plant has enough stuff going on around here in the soil, one presumes, it opens the doors into the root system. But this happens just at the root tip. And so in come all the bacteria and the fungi, having a grand old time. Isn't this fun? And then the plant shuts the door and sends out an oxidizing agent that starts to cause all of those bacteria and fungi to lose their ability to hold those nutrients inside their bodies. So here's the plant just sucking down all of the nutrients that it needs. The microorganisms are kind of going, well, this is getting kind of unfriendly, guys. You're going to wipe me out if you don't let me go. And so they move up to the first root hair. And there's got to be an escape door. Nobody's demonstrated where it is, but there's got to be a way to because all of a sudden all these organisms are appearing on the outside, and they cruise on back down to the tip, getting fatter and stronger, pulling in the nutrients that the plant took from them. And so they can run around to the bottom and this has got to be like a 3-year-old behavior again. because just like me, they're running back. Oh, that was so much fun. Again, come on, do it again, do it again. See, I've filled up on all the good stuff. So the just over and over and over, for as long as we treat our soil like it's something living, because it is a living entity. Yep, it's all about the biology. So so there's one thing I just can't not thinking of to
All
straight.
right, I got a great question I've always wanted to ask you. And I don't think there's very many people that really know this, but you actually came from the marine biology side of
Yeah.
things before you popped into the soil food web. So talk to me. How did you transition from water to soil?
Well, it was fairly easy because um, there's no money. At that time, there was absolutely no money to to rely on being able to get a job once you finished that next 4 years of your life doing a PhD. Where would I go to find a job? And if I was going to have a problem finding a job, maybe my husband was as well, because he was working in aquatic aquatic nematodes. And so we were both, this is not, we don't want to stay here in the world um, of aquatic.
Starve to death world.
Yeah, yeah. And yeah, so it wasn't gonna be easy. And then we had decided that we would automatically go to the school that gave both of us research assistantships. And the place that gave us good, something that we saw we would really enjoy was from Colorado State University, where I worked with in soil microbiology, which is, boy, that's a shock. When you come from the aquatic system where everything's all nicely mixed up and moving around, and you don't have all this glop in there of all this dead organic matter and layers of sand and silt and clay, and it's just like you start dealing with soil and there's so many problems with trying to see what's in there. So I had to do quite a bit of work the first 6 months I was there to kind of catch up with the other students who had been in marine or had been in soil science, soil microbiology. I had a little bit of had to work at it pretty hard to get caught up to everybody else. But then, working with um, Dr. Coleman, as well as my major professor, really turned things around. And they had Dr. Coleman had a grant from the um, USDA, I believe it was, and they wanted to do the food web structure in all these different biomes and be able to define exactly what the biology was. And one part of what they were interested in was what are all the organisms down there in this and all of these different biomes. And so they just were doing cataloging all the different organisms they saw in those places. To me, that was a little bit, oh, really exciting there. So when I finished up my PhD with Dr. Donald Klein, uh, and in soil microbiology, um, they were looking to the people in the natural resource ecology lab was looking for people that wanted to really dig into and start to understand how things worked in that rhizosphere. You have all these organisms in there, must be doing something. Mother Nature doesn't just leave things doing their stuff without there being a darn good reason for them to be doing that. We just had to figure out what they were really doing. And so, doing a number of different experiments, looking through the microscope. And so that was a really a very important part of what I was doing was to discover a very simple and easy way to determine whether you had too many fungal hyphae, whether it was just about the right amount or it was too low with the fungal hyphae. Starting to, oh, and another thing that we found was quite recently was when you look at bacteria, their biomass has to contain 5 carbons, 1 nitrogen. When you look at the foods that bacteria eat, generally, they have 30 carbons, 1 nitrogen. So when a bacterium eats anything it's supposed to, that it likes to eat, that means it's being stressed for the lack of nitrogen. So what do you do as a bacterium? How do you get rid of all those excess carbons? You blow them off as CO2. Every time you till your ground, and bacteria can tolerate being,
Tilled.
tilled. Yeah. So
Tilled,
they,
tilled.
yeah, yeah. But the fungi, they're all sliced and diced and crushed, and, it's really bad news. So all of that extra carbon, all those extra food resources that the fungi used to be able to control, they're going to be chewed on by the bacteria, which is even more of your organic matter getting blown off into the air because bacteria are inefficient on carbon. Carbon is not what controls what bacteria do. So what do we have to do to bring back that ability to sequester all that carbon in the soil? We have to grow fungi. So fungi have a carbon and nitrogen ratio of, maybe something like 60 to 100 carbons for every 1 nitrogen. And so when a fungus is eating that, any kind of food that it's got, it's in that range as well. And so the the fungi are going to take all that extra carbon and put it down on the inside of the hypha, storing that carbon there. And so we increase the amount of carbon that can be put away, not blown off as CO2. It stays in the biomass of the fungus. But every time we slice and dice and crush those fungi, think of all the massive amounts of carbon that are going to be converted into CO2 by the bacteria. So because we don't understand these things yet, we allow people to get away with these kinds of behavior. I hope we convince them not to be so foolish in their uses of their natural resources.
That brings up another great question, and that is that I remember you talking on this way back when about fungi's ability to, they have a hyphae that goes out, finds a food source runs out, the animal retreats, but then they use the whole hyphae as a storage tube.
Mm-hmm.
For storing all kinds of things, including excess carbon, correct?
Yep.
So it's probably even 1,000 or 10,000 fold even greater when you start to destroy something that is fungally dominant, is relatively virgin. Maybe it's been recovered or followed for 3 to 5 years, but that's where you're really creating these negatives that are almost incalculable as far as municipal costs.
Because most people in soil science don't look at the full depth that the root systems of the plants are going. When you look at old growth anything, those root systems will go down 250 feet. And all that time, all those microorganisms around it, very fungal dominated under those conditions. And that is going to keep massive amounts of carbon sequestered in the soil. We go in and we cut that forest down, and all of that carbon is now going to be released back into the atmosphere as CO2. And soils people just don't understand those kinds of interactions. They look at just what's in the top couple of inches. Just couple, it was probably like a week or so ago that they opened an exhibit in, I believe it was Iowa, where they had the above ground plant material up here, and they showed the distance that those root systems were going down something like 45 feet. And those are just kind of average, common, everyday plants. And yet, soils people have always told us that the root systems didn't go down more than maybe 2 inches. See what's wrong with our mental outlook is we have people who just refuse to go in and find out what's really going on.
So yeah, you've heard me often bitch about soil dogma. You know, the soil science teachings of chemistry and physical properties, and not even looking at the biological part of it.
Yeah,
And,
biology isn't part of it from their point of view.
yeah, and it's crazy that here we are in the year 2023, and that's still being taught. You know, when the rest of the crowd knows, no, if all three are the way you should look at soil: physical, yes, sand, soil, clay, and organic matter. It's important. Chemistry, absolutely important. But without the biology, guess what? The chemistry can't be released or uptaken. So, yeah, you know, that's one of those things, Elaine, that frustrates the hell out of me. But, you know, we won't focus on that. We're going to stay positive here.
Well, it's
Um,
like it hasn't been too long ago that somebody sent in just a nasty letter about how we tell people that there's all this organic matter in the soil and that your plants are growing, they must have more than 5%, 10%, 100%. Well, my favorite place to grow plants is in 100% organic matter. I don't need sand, silt, or clay. Sorry. That's not what's giving, allowing those plants to grow rapidly. So when they're saying, well, the soil can't have more than 1% organic matter. You just can't get enough organic matter into that soil. And I'm like, where are they coming from with this thing? Because, and what I finally realized is they've only and ever paid attention to agricultural chemistry done with toxic chemicals. And so you apply that toxic chemical and you've just gotten rid of all of the bacteria and fungi, protozoa, and nematodes. And now there's no way to hold those nutrients in the soil and they wash out of existence. You're just putting high levels of all of those materials into the water and causing us all having to drink water that is toxic. We eat plants that have a layer of toxicity around them. We give them to our children. We get, they're eating good, right? Well, not when you understand that you should have no need to put on inorganic fertilizers or pesticides or anything else. You want to grow the plants in a condition where you don't have to put on those toxic chemicals. And that means you can't be putting on the toxic chemicals, but you have to have the biology in those places. Soil does not begin until it's at least 5% organic matter or higher, all the way to 100% organic matter. How about those where we grow most of our agricultural crops are in dirt?
Yep.
There's not even 1% organic matter, 0.01% organic matter. And soils people have told me to my face that that's the soil that you should be working with. It's not soil. And how can you pretend that that's soil?
No, again,
Okay,
if it,
I have to jump in here. I don't think you had enough inflection on how bad that word is of dirt.
Dirt.
thanks, Ken. You're
A
right.
little bit of growl.
Yeah, you got to put a behind that.
That's right. It's one of
No,
those things that I just can't believe that people still tell me this. Where are their professors? Have they not looked at a recent book Have they not read the recent literature that shows how critically important it is to have that carbon or have the organic matter up above
Well,
5%?
Elaine, it's like these farmers, right? They're just so set in their ways that they don't get it. And they look at their soil, and if it runs between their fingers, they're happy. And it's like, no, you want aggregates. You want organic matter in there. You want all of those biofilms and those microenvironments. And that's how you're going to grow good corn.
Yep. And if you have a year that's going to be droughty and you don't have some way to form those aggregates and hold that water, you're going to lose your crop.
Mm-hmm.
And whose fault is it then? Don't want to be paying these guys subsidies.
And that's
They're
what
idiots. And
they go out and get crop insurance.
Mhm.
And guess who pays for that? The taxpayers. It's just a circle of insanity, totally. When all they have to do,
we gotta stay positive now.
Oh, thank you. You're right, Av. Keep me in line.
I'm gonna ask and once again, always, going back, giving thanks for. Many of us are going to be familiar with the ecological succession because that's one of the things you talk about in the course. So and now you're a strong advocate of no-till systems. So how can you give us some practical ways of maintaining that fungal bacterial ratio for our desired crop in a no-till system, so that it's not going to progress to that old growth forest, So how do we do that?
Well, you don't have to worry too much because going from grassland to old growth forest is going to take you a couple hundred years. So not something we're typically all that worried about. But, we do want to make certain that we get the proper biology. And so you're going to be looking through the microscope to see what's in that sample. And then, what's the minimum that those organisms should be held at? We want 136 micrograms of bacteria. We want 135 micrograms of fungal biomass. We want to see somewhere around 10,000 protozoa per gram of soil.
And
want
this would be like if you're growing row vegetables or something like that, or
anything. These numbers are anything. You have to start at these numbers and make them better during the lifetime of that plant, because you put them back there at 15 parts per million or 8 parts per million, you're just not going to get the system working that rapidly when you start from such a low place. So if people are, well, I don't want to do anything other than just let it go, well, then it's going to take you 2 or 3 years to get to a point where you have at least the right amount of biology in your soil so that you can develop a really healthy, good soil. So once you've reached above that minimum level, now, and hopefully you've got way more of those organisms than the minimum. So, okay, so part of your work's already done for you because your microorganisms already were getting it together. So now it's going to be based on what is the plant that you're trying to grow. And so really early in succession, you have disease-causing bacteria, disease-causing fungi, not a really good, healthy place. But you want, when you see that that's where you're at, and what you're going to try to grow are, say, tomatoes, then you want to start improving that biology in the soil by adding compost, properly made compost that's got the right biology in it. It's got the nutrients in that organic matter, and will get those nutrients out of that organic matter and give it to the plant. So when you see that what you want for tomatoes, you want that ratio of fungi to bacteria to be something like 0.75. So you now get that balance, a little bit of bacteria, a little bit higher amount of bacteria, but still a good community of fungi in there. And now the protozoa and the nematodes chow down on the bacteria and the fungi, and everything starts cycling really well. Let's say you wanted to grow apple, an orchard of apples. Well, now instead of a fungal to bacterial ratio where the bacteria are winning, we're going to cross that line of 1-to-1 ratio, and now we are in a system that has a reduced amount of bacteria, and the fungi are up there at 25% higher than the bacteria are, and you're setting the stage to grow that plant. So it is kind of a two-step process where you want to be aware that as long as you got good sets of organisms, enough, great. Now you can start making it something that's going to give you outstanding productivity.
So, like, if we were to take, and many of our viewers are cannabis growers, if we look at that plant and growing it outdoors, maybe 20 weeks, maybe 24 weeks, would you feel very comfortable making the same compost tea that you're using for a seedling as you would be using in late flower, or compost extract? Do you feel like do you need to change your amendments during that life cycle of that plant if you started with a biocomplete compost?
Yeah, that's an interesting question because we haven't really seen those kinds of data. And where you're going year after year looking at data from any particular kind of plant, the 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 food web interaction, the ratios of fungi to bacteria to protozoa to nematodes, we don't really need to change them as we're going. We know that there will be a shift as the plant is finished accumulating and collecting all of the nutrients that it needs to take to grow a seed, a flower first, and get the flower, with the pollen, and now we're starting to grow the fruit of that plant. There is a shift, higher ammonium being produced. You got to have a lot of energy being produced to grow that material. You've got to have all the nutrients there in the proper balance. But I think that's not really a lot different from where they were ending up in that growth period, and now by putting on the organisms. So yeah, here's where we need some more funding.
Right. With another grant, it seems.
Right.
And you would feel very comfortable because many of our growers will use weekly applications of compost tea throughout the life cycle of the plant. Do you think that that's challenging on the plant in any way?
Nope, I haven't seen that challenging as all. What might get in the way is they'll get the soil too wet. And now, especially if you're in a pot or something like that, the bottom 2-3 inches are going to be totally saturated with water. So, punch a few more holes in your container so that it's going to maintain. If you're growing in a greenhouse, you probably don't have to worry. It's when you're growing outside. Now, how are you going to tell whether, okay, you stick your finger down there and through the soil, and okay, is that really wet? Is that really dry? And you want moisture. For cannabis, you want it around 40 to 45% moisture.
So, of course, we're always looking for those secondary metabolites for our cannabis plant, right? And are there little tricks that particular functional groups that you're trying to get more of to produce either higher terpenes, flavonoids, cannabinoids? Is this something that you're wanting to do more research around, or is this something that you already know a lot about?
I know a little bit about it, and it's kind of like the money for doing these kinds of studies just seemed to disappear 2 years ago, maybe 2.5 years ago. Um, that yeah, we were getting some interesting results from making certain that the soil was more fungal dominated than we had thought, looking at the plant growing in the first 4 to 6 weeks. um, then we started looking later and later and later, and it became very apparent that at least certain for some types, some of the groups of cannabis, they definitely needed something way more fungal in order to produce those flavonoids. Now, that's about as far as I've gone on that particular question, because sorry to be need funding. It's necessary to be able to give those little greenbacks to the grocery store and be able to eat.
So interesting that we ended up on the subject of compost yet again. And what I would like to recommend to the audience, and anybody listens to this in the future, please send out your compost to a soil food web consultant. Verify whether it's bacterial or fungal. If it's fungal, then yeah, you should be using that every week through flower, because as she just stated, that's really important. And the only way to get that fungus in that short-lived plant that's not planted in the ground is by you doing it. You're the driver. So make sure that you're doing everything that you can to get that stuff in. Soil food web test is not expensive, people. So there's no reason why you shouldn't, because it's the roadmap to where you're going. Like if you're going backwards every year when you send in your tests, it's getting worse. Well, now you know that. Elaine, I don't know about you, but that's always been my biggest frustration is how many people come to me, oh my God, I make the best compost. I've been doing this for 30 years. I know what I'm doing. And you put it on the microscope and there's nothing but bacteria. Again and again and again. And they're like, no, mine grows plants. Of course it does. It helps. It's compost, but it's not fungally dominant. That's always been the big challenge. And, while we're on the conversation of compost, have you noticed the literature coming out nowadays describing compost in stages in succession? So there's POEM, particulate organic matter. Then on the other end of the scale is MOAN, mineral associated organic matter. And it starts as either ground or unground organic matter or soil organic matter, SOM. So where do you think BioComplete comes into that? Somewhere in between POM and MOAN?
I think it covers all of that. Just take all of those, whatever, all that gamish of different kinds, and everybody's definition is a little bit different too, which drives me insane. Because how can I respond when there doesn't seem to be some arbitrator saying, no, that's not the right definition. Just take the organic matter and look at it. I don't think we need any more. But I'm somebody I really want to get done in the most efficient and least time consuming fashion. So you just need a lot of organic matter, and that should be enough.
Right. And again, though, I'm just trying to push the definition because compost is a process, not a product. And I think that's the biggest problem with people is they hand me the bag of stinky, nasty compost and they say it's compost. I'm like, no, that's anaerobic soup. That's not compost. At least with POEM, you might not smell good, but it's not it's not anaerobic. It's not going the wrong way. And so again, if we're going to ever move people to a place where they understand what BioComplete compost actually is, we need to start creating definitions for the various successionary stages of composting, because that's really what's going on. And I think you set a bar that's beautiful and it needed to be set because nobody understood compost, not all composts are created equally. They come from different ingredients. Like you said, yard waste is not compost. It's ground up organic matter. That's all it is. And it heats up and blows off nitrogen and methane. And so that kind of in many ways is, in my mind, the wrong direction. And so
Sure, because you just described anaerobic waste products coming from those organisms. Those aren't beneficial organisms you're growing in that putrefied organic matter. Call it what it is. I want to make certain that you don't have any anaerobic conditions in your composting process, because the bad guys grow in reduced oxygen conditions. And when you smell those bad smells, that's telling you, that's Mother Nature saying, hey, pay attention over there. When it stinks like that, it means it's anaerobic. How do you
right.
get rid of the anaerobics problem? You aerate it. But now that you have killed most of your good guy organisms off because it went anaerobic and you didn't pay attention, now you're going to have to start generally at ground zero with that carbon, the organic matter that you have. I don't ever make the whole next pile out of that woody material that's left over when you go anaerobic. You've always got to probably no more than 5 or 10% of that spent waste material should be used in the next pile. But it's not like you have to throw all of that pile away and forget about it. Just let it have some time to relax and go aerobic for a while, and go ahead and use it then as it gets older, or should I say mature.
So maybe we should define compost as a smell. In other words, if you know what it's like when you have really good compost and you smell it, you're like, oh, this smells like forest inoculant. Maybe that's the way to say it. Like
there are just too many people that have nasal problems. They've been sniffling their whole life.
Or they've never smelled a good compost.
That's the other thing. We have to have an institute that everybody has to come to, and as a small child, you must memorize the smell of good
There you
compost.
There you go. Or maybe the scratch-ons. Like, remember the old scratch-ons perfumes? We could get scratch-ons that you put in an envelope and send out to people. This is what it should smell like. If it does, then you're okay.
Yep. There's just I like going with the biology, looking through the microscope, because you come to the foundation courses, you get to foundation course number 4. We're gonna teach you the qualitative method of assessing compost. It's gonna give a little bit of quantitative in there as well, but as long as you have the right balances, you don't have to get all crazy about finding every little tiny itty bitty bit of fungi in there. Just do your best with that scan across through over the sample, and it's good enough typically, as long as you're honest with yourself. And that's
Right.
Sometimes people, if that's close enough, that thread of blue jean material coming off my clothes, that was a really good fungus. No, sweetheart, when it's orange in color, I don't think that's a fungus. The pink and the blue and sorry, no, not
And I
fungus.
suffered that one. I brought you a sample, and I'm like, look, I got blue fungi. I'm so excited. You went, baby, that's your blue. Broke my heart. Broke my heart. But I remember when you put me in that group of students that you had on retreat that summer, and you said, Leighton, go up and observe these guys, listen to what they talk about, and follow the directions that they're giving you on how to learn how to use the microscope. And one of the things I noticed pretty quick on, because every time one of them said, oh, look I got a great one, I would grab some of their sample and put it under the microscope and look at it and try to figure out what they're looking at that made it so good or so bad. By the time they left, I'd realized that if they were in a good mood, they saw a ton of good things. If they were in a bad mood, they saw a ton of bad things. And it wasn't necessarily correlating with what they were actually looking at. So it's so true that you have to, really be honest with yourself when you are looking under the microscope, because otherwise you'll just chase the good things and you'll ignore all the bad things.
Yep. Yep. And the whole purpose for us doing these things is to figure out the truth about
Mm-hmm.
what you have to help get along and be able to resist the diseases and the pests and the problem organisms, where they can get the nutrient they need. They can store away the amount of water that they're going to need for the whole entire growing season. Just it appalls me when you see people totally anaerobic fields, compaction layer at a 1/4 inch, and they
Oh, yeah.
won't. Oh, yes, and they won't fess up to the fact that their water is running downhill.
Yep.
It rains the next day, and you go out and you say, "So where's this little river coming from?"
Or black snow. I'm sure you've seen black snow. That's another one. It's like, oh my God, don't you get it? The little organic matter you had is now dust in the wind.
Yep, it's a goner, or it's in a better deserving place.
Hopefully.
Yeah.
It doesn't hit the neighbor and keep going until it hits the forest, right? Well, maybe good for the forest, right? Maybe good for all the wind blocks.
Yep. Accumulating someplace other than
Where we
it
need.
was hopeless, absolutely hopeless, and just bad things were happening. Yeah.
So I'd like to take us on another fun little journey. The work that you did over at Chernobyl. You sussed out the fact that isotopes could be stored in mycorrhizae fungi. And then those damn Russians drove into friggin' Chernobyl and dug in and set up shop and stripped every one of those friggin' goddamn nuclear alarms because they broke the fungi. So can you tell us a little bit more about what you found during that work, that incredible work that you did?
We found that some of the plants growing in the field that had mycorrhizal fungi growing inside of them. And so maybe I need to talk a little bit better about what that um, process was. The root system on the carrots. Yeah, so you have the carrot itself, which is orange and fairly chunky. The green top up here, but then hyphae growing out from the root of that carrot. And every one of those samples that we looked at, where there was mycorrhizal fungi coming from one plant and colonizing the next plant and colonizing the next plant and the next one, um, none of the actual carrot root had the
Radioactive isotopes.
radioactive materials. And so where are those radioactive materials? And we took the root systems, the small, on the sides, we took that material off and looked at it, and whoa, that was, guess what? That's where those radioactive materials were being stored by the carrots. And so, we didn't have money to go looking at more kinds of plants that had, were they, like blueberry. You need to have a um, you need to have a mycorrhizal partner to that as well. And so would it be the same? Would blueberry, blackberry, whatever, would it have those problems in the berries? And as long as they had mycorrhizal fungi, I believe they would. Now, we didn't actually look. Where we had the carrots and they had been treated with some sort of weedicide on it, and it killed the fungus, then the root of the carrot had radioactive material in it.
Amazing. Just amazing. you know, and it just proves that what you discovered there was in fact true when they went in and drove their tanks and ripped the whole friggin' forest up because they released all that. So obviously a lot of other plants were doing the same thing, is basically saying, no, 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 I don't want that isotope. So you go bury it over somewhere else and in someone else's backyard. What is it, NIMBY? Not in my backyard.
There's been a group in England that has been going around the world looking at these different kinds of interactions, and their data fits our data perfectly.
Nice.
So it's, you know, it's getting out there to the wider era, to the wider people
Audience, yeah.
audience. Yep. And it's we've got some work that we want to do this not this summer. It's a little too late. The money got in too late. But next spring we'll be looking at that kind of situation a bit more in depth. So
would it be just isotopes, or would it be all kinds of different things like heavy metals?
Yeah, we plan on doing work with heavy metals, all kinds of different toxic chemicals as well as the yeah.
You know, it's funny, but again, Elaine, you have inspired so many people across the globe in so many different directions. And again, I want to thank you from the bottom of my heart, because it's given me the ability to live a life that feeds my soul. I'm not chasing money, but I'm doing good things that make me feel good. And I don't know if you remember this project that I had with James Satillo. It was the Chattanooga auto dealership. And I came to you and I said, Elaine, I had the soil tested. It's got a tremendous amount of coal tar. And you said, all right, you need Pseudomonas. And I'm like, okay, well, how do I make Pseudomonas compost? And you warned me. You said, Leighton, if you're sick, you're going to get really sick playing with this stuff. But if you do it appropriately, Pseudomonas will, and you respect it, the Pseudomonas won't hurt you. And you gave me a recipe. I made it up. I forget, I was still at Rodale or now, but I made this compost and sent them down the extract. And sure as shit, they cleaned it up. So, I mean, again, bioremediation is like going to be one of the real big issues if we don't crash into the 2030 agenda, all this shit, right? But there are so many Superfund sites and so many dirty areas. Even, you know, just a regular farmland has so many heritage freaking chemicals, atrazines, and so, you know, this is really like the one of the most important areas that we need to look at in the future, is that through composting correctly and appropriately and using materials that we know do have these specific bacteria that can break these metals apart, or these compounds, degraders and decomposers and solubilizers. We can make amazing changes, and in a short period of time. I mean, we did that in 1 year early. It went from horrible levels of coal tar to very little in the course of about 18 months. And so, have you been playing around with any more of that type of bioremediation work?
We've got a proposal that looks like it will be funded, maybe, hopefully, please. And if
Fingers crossed.
yeah, how many ways can you cross things? You
Do you remember Twister?
So if we get that funded, then we're going to be using compost as a filter for all that contaminated water to go through. And should be possible. We should be able to manage it. You got to realize that, gosh, I'm not going to remember the correct number of digits in here, but you know, it's like 6,000 new toxic chemicals are released each year, on top of all of those that we've already had. So how many, you know, since World War I, how many years has that been? And we've been happily producing new and different toxic chemicals that are soluble. They move into the water, they move into your wells, your lakes, your oceans, and no one seems to get upset by this. We have to stop trying to grow our own food using toxic chemicals. It's just such a mental, what are you thinking about? Um,
Money.
and that's all that matters in this world, right? Is money. Um,
It appears like that sometimes, Elaine. And the reason I asked that is because a good friend of mine, Leaf, has gotten a job down in Oklahoma, studying mine waste. So this is a lead mine, and the wastewater that's coming out of there is horribly toxic. And originally, they set up a series of bags filled with spent mushroom substrate only, and filter the water through that and change the bags periodically. And they have had a tremendous amount of success with just that. And I believe he's hoping if he gets allowed to continue in the program, to start introducing more compost, because we did a study with Furman where he reached out to me. He had shown that the ability to break atrazine down to secondary and tertiary metabolites using white rot fungi, but the metabolites were just as toxic, but they didn't last as long. So he wanted to eliminate them. So he asked me if my BioComplete compost would maybe benefit it, and I was like, heck yeah, why not? It can't hurt, right? So
there,
they did a
there,
series. Say
there have
again.
been, have been papers on atrazine showing that in a very short period of time, they could be completely decomposed.
Yeah, yeah. So when they added the compost with the white rot fungi, gone.
Yeah.
So again, it drives back to the fungally complete or fungally dominant compost as the solution to the pollution.
Yep.
It's not dilution. It's to remove it.
Right. It's the solution. Yeah. So yeah, hopefully we'll get funded so we can be doing that next piece of work on all of this. But it does, it's something like 85,000 new toxic chemicals have been have been developed in my lifetime.
Oh my God, that's horrifying.
Yeah, so we've got a
We
lot of work to do. We have massive.
pull the words right out of my mouth. We still got way more work. We're creating more work than we're getting accomplished. So where is the project that you're hopefully getting funded for?
It's in Oregon.
Okay.
Yep. So it's just a hop, skip, and a jump away from the new lab.
Nice. Is it a super fun site?
No.
Okay.
Yep. It's from specific kinds of neighborhoods, and they really wanted to look at not so many of the things that were enormously problematic, but how about all of the little things that come through in a town of small size?
Got you.
How
And so,
expensive would it be? Right now, they've got their own, water factory or water.
well,
Yep. And so, how if it got contaminated, how rapidly would we be able to get rid of it? How do you deal with that kind of issue as well? Where's it coming from? Well, today you can say from people's front lawns
mhm.
and their backyards, from the park down the street, the school grounds that your kids are going to.
The
Uh,
dry cleaners.
yeah, and we can decompose these things. We don't have to sit by and just go, "What are we to do?" So hopefully we'll be able to speak to people who will understand
Fantastic.
and really start working on these things. It takes people like you and me doing the research. We need to, yeah, we need to get the politicians involved, and that's another aspect of Soil Food Web School is that we're making contact with a lot of the people who are really serious about working on uh, dealing with toxic chemicals, exiting them from our lives.
Well, that's great news, Elaine. That's really, really great news because it's, every time I've ever tried to get involved with, these entities like governments or the environmental science companies, they stonewall you, they shut down. So, and it's going to take someone like you with your clout and that many years of doing the hard work and the hard research to finally turn them around, because it's basically like trying to turn around an aircraft carrier. You can't do that shit on a dime. So, kudos to you. Well, we're at 1.5 hours. I want to be, genuine and thankful for your time. We have 12 questions coming in. So why don't we go ahead and get the audience's questions answered, and if we still have a little bit more time at the end, we can chop it up a little bit more.
Okay.
That sounds great. Now, some of this stuff I'm not going to be able to pronounce worth a dang, Elaine, so I'll just let you know now. We'll start with perpetual here and bring this up. And here is a good point to repeat my questions from last week. Why add too much fungal microbes when food crops generally do best in a bacterial dominated soils, 0.6 and 1:1?
It's really because we haven't paid attention to the fungi and what they can do. And so most people are moaning and groaning, and how are we ever going to get rid of all of these toxic chemicals? And they're only using bacteria. It's the fungi that are the real, those are what really take care of those complex structures, break them apart, and turn them into the constituent of an amino acid or some other carbohydrate or to CO2. And so pretty simple to answer that question, because we've got to have the fungi included in here if we want to do a complete cleanup.
Elaine, can I ask a question on that? When you do a soil food assessment on an F to B, so fungal to bacteria relationship, is mycorrhizae included in that, or is it just saprophytes?
It's the mycorrhizal fungi are included, but what we're included in is in the soil. We miss the part that's in the root system. So we're underestimating the actual total amount of fungal biomass that's present. But
There you go. Endophytes, right?
Yep, that's right. Yep. So when you look at ectomycorrhizal fungi, they have massive amounts of strings and threads inside the root system of that on the outside, in the first
Cell wall.
layer, cell wall, cell membrane. So it's, yeah, we do the best we can.
Well, hey, you built a foundation, and now everybody wants to build a skyscraper on top of it without respecting the foundation. So shame on them. But anyway, next question. Sorry.
Oh, no worries. No worries. Anything against using bacterial fertilizers like urea?
Urea really isn't a biological fertilizer because it's usually been taken through a processing plant. I don't remember exactly what goes on. You need to use urea carefully. What are the salts that are still in that material? And so I would probably back off significantly. Do a set of experiments where you know exactly how much harm that urea did to your organisms in the soil, the organisms in your compost, so that you know what you're doing when you add that material to anything.
Which makes a lot of sense.
Yeah, don't be a moron.
Keep the balance, people. Keep the balance, right?
Right.
You don't empty the whole salt shaker on your meat, right? Just a couple taps.
Yeah.
Anyway.
So how important is the time of day for application of extracts, teas, and watering plants?
Well, we thought that, it'd be very important to have to keep track of when you were applying. So we did an experiment where we went to a pivot that was, I think it was 135 acres in size. And so we started very early in the morning applying a compost tea to the potato plants that were in the system. So at 4 a.m. we started and just let it chug along. We figured that when it hit high noon, that we weren't going to have very many organisms left
Hmm.
because UV is supposed to kill these organisms. And so, okay, we're going to find out whether that's true or not. And so the the arm went on going on along. Let's see, I think it was dark at like 8:30 in the evening. So we knew when the sun went down, we expected to see an increase in the biology being applied there. And so went on through the night and finally hit the next morning. We finished up at about noontime come the next day. And we went out then and took samples throughout all of that area,
Hmm.
knowing that how much sunlight was being hit, that was hitting those organisms in that soil. And what we discovered was it made no difference.
Wow. Wow.
Interesting.
So that was one full pivot. It took 18 hours to get all the way around, and you sampled like an hour all the way around until you got back, whether it was at night or in the middle of the day, and it didn't matter.
Yep, and it was a slower pivot. It's 135, so a little bit slower. And so it was out there for 18 hours, I think it was. So it included the next morning, and we were really surprised at
That's
those
Mhm.
results. Um, so a lot of it is that we were putting the application on pivot that already had plants fully grown in it.
got
And
you.
Mhm.
so as this is going on to those leaf surfaces, they experienced a lot of shade,
Got
preventing the damage that we expected. Now, if that had been bare soil, I'd expect something very different. But we've never gone back and done that work. What if all of your water is immediately absorbed,
you.
infiltrated into your soil? Then I wouldn't expect. So it's only when you've got compacted layers and the water cannot infiltrate, that's the only time UV is going to cause you harm.
Mhm.
Thank you. That's beautiful information. Beautiful information.
Yep.
Definitely gold bars on that one. Definitely. So I don't do compost. Is the worm farm good enough?
And I'd say it depends. Um, what are you putting into your worm bin? Are you reaching the um, the concentration of the bacteria and fungi and protozoa and nematodes? And there are cases where absolutely you're getting really good compost out of a worm bin. It's probably a little bit harder, but to get a really good food web going in a worm bin, but if you achieve it, it's probably better than a thermal compost. So it also depends on what you're trying to grow. And,
Mhm.
so it's a long list of possibilities. Check it out. Make a thermopile and make a worm bin and see which one you like to do work on. Some people really like the idea that you're going to put this plant material together and in 15 days it's going to be finished. And you don't have to fuss with it until, you know, a year from now when you have need for more compost. Worm
Mhm.
bin, you've got to be there to put in the food in that worm bin and make certain that every 3 days you put another increment in there because you don't want any fruit flies, fungus gnats, or other disgusting little creatures running around in what you're going to put on your soil and
Mhm.
try to prevent diseases and pests and all of that. So up to the grower, really. Just make sure that you have the proof that you've got the bacteria and fungi, protozoa and nematodes up to their minimum requirement levels. Maybe you want that you're trying to grow pumpkins, so you've got to get that fungal to bacterial biomass ratio up a little bit higher. You need the fungus just, right in that 1-to-1 ratio is what you
Mhm.
really want to look at. So it depends on the crop, depends perhaps a little bit on a number of different things that you'll very rapidly find out about if you don't have them.
Well, and that's the importance of doing side by sides and testing what you've got against what you're trying to create. But it kind of leads into the next question. Got to get that temperatures hot. Now they're talking thermophilic compost. We talk a lot about not taking it to the extreme temperatures and not knocking the biology back. How do you feel about that?
That's 131 degrees Fahrenheit, which is the uh, cutting point. For below that, you're not getting rid of the disease-causing organisms. If you hit 131 Fahrenheit, then you're gonna be killing most of the disease causers. But it's got to be hot enough, long enough for a full 3 days. You must be above 131. If at any time the um, temperature in your
Compost.
thermal compost, thank you, um, thermal compost, um, we want to we want to have that temperature up high enough, long enough, so that you can be assured that the disease-causing fungi and the disease-causing bacteria, protozoa, nematodes have all been dealt with.
Hmm.
Um, you want to turn it at that point. So now you're getting the hot inside moved to the top. What was in the the turning is got to be done properly, and then you have to get to above 131 degrees Fahrenheit for a full 3 days for a second time.
Hmm.
So you've got to reach those temperatures. If at any time you drop below 131, you're you need to run out and get some blood meal really fast and dig
I
down
think
into the middle of your pile and put out a couple of handfuls of blood meal into and that usually does the trick because it just drives me nuts. I'm only 3 hours short of having perfectly good compost. What can I do to keep it up there? Okay, so that works. And then you've got to change your recipe for your next pile because you
Hmm.
better put in that additional nitrogen. So yeah, it's with thermal materials, does it hurt things too much if you get really hot? So let's say you get up to 170 degrees Fahrenheit. Um,
Hmm.
you only have to You only have to have it up at that temperature for a very short period of time. It's not dropping into my brain what that is, but it needs to be held at that level. If you leave it too long, because the organisms are growing so fast and they're utilizing so much oxygen to keep the temperature up at 170, 171, something in there. See, 71. That's why I chose that. Um, birthday party time. Uh,
Yeah, yeah.
so you have to make certain that you turn right on time because
Hmm.
otherwise it goes anaerobic. Um, Alcohols are only produced under anaerobic conditions. And now you've got something that if it reaches 180 degrees Fahrenheit, that alcohol is going to burst into flames. And
Mhm.
from then on is a really horrific scene because how many people have the ability to put out a fire in their backyard or in their barn or wherever they are? Most of them lose a significant amount of equipment, or
Yeah.
yeah, there are definitely problems. So try to keep it down to lower levels. Yeah, so hot enough, long enough, get your recipe right.
Yeah. Which really answers the next question as well. So anaerobic teas are bad. And you basically just answered that question.
As well as it's the anaerobic organisms that are the diseases and the pests and the problem organisms. Yeah, there's a few that are like wimpy diseases. They form little dots all over your flower petals, or the stem of your roses are all anthracnosed out, and you, but they're not going to kill your plant. So really, are they disease causers? Not
Mhm.
really. They're just not something that you want on your treasured roses, because then they
Yeah.
don't look that what you want them to look like.
And we want pretty roses, damn it. Come on.
No, yeah.
It's
Isn't
for
that
your
good?
birthday.
No dots.
Yeah.
Where the
Elaine,
roses were they?
I just want to make sure that you're not totally opposed to EM1 or a well-designed fermented plant extract
You
necessarily.
you have to know why those things work. And so if you're going to be going after the pests and the diseases and those things, it may not do you much good to make something that's anaerobic. Well, if there's some really toxic chemicals being produced by that anaerobic brew, and now you put that into your compost pile, yeah, you'll kill some things, but now you've got to deal with the fact that that's a toxic chemical.
Mhm.
And I'm not really too interested in putting that next to my lettuce. Thank you.
Why not? I mean, come on. What harm could it actually do?
Absolutely. Other than you end up throwing up all night.
Yeah.
Hey, it
Oh,
happens
I
every
went to
once in a
Mexico and I
while.
drank the water. Whoops.
So
So
just
I'm not even going to
know
try to pronounce this, Elaine. So if you want to look at the screen,
Acetobacter aceti usage in composting. Uh, I I guess I haven't kept up on reading on this thing, but I don't. Why would you put in um, something that? Yeah, I don't really know what its uses for. So I'd have to go looking in Google and copy the Acetobacter aceti and do some reading. So that's guess what I'm going to be doing as soon as I get off.
You're going to
well,
your
you're going
birthday
to be
party
having
first.
cake.
Yeah.
Yeah. Yeah. Cake first. So another one from Ghetto is I'm using spent mushroom substrate. It's beneficial for fungal composting.
Oh, sure. Yeah. It's a really woody material, so I would put it in as woody. It's the wider C to N ratios that will grow some really good fungi. Now, you want to be a little bit careful and make sure that it's not, um, like growing some disease-causing materials, because it's been sitting out in a pile in the middle of the forest and nobody's done anything with it for the last 3 months. And
Mhm.
oh, you could be growing some really interesting anaerobic material there. So know what you're putting out. This is why you use a microscope, so you can see what's there and know that looks pretty good. Or, whoa, I don't think I'll put that
Whoops.
next to any of my plants.
And that's the key, is using the microscope so you actually can see what's there, right?
Yeah, exactly.
Would mushroom substrate add carbon in a form that would be readily uptaken by plants?
No.
Okay, that was a quick answer.
Well, it was a nice specific question, and it's yeah. There are other things that are going to produce materials that are uptakeable by a plant, and
Mhm.
just fungi chowing on it. No, no, it'd have to be
Okay,
invoking enzymes of some kind.
okay, and then our Pseudomonas amoebus.
Pseudomonas is a genus of bacteria. Amoebae are a broad group within the protozoans. So amoebae eat bacteria,
Okay.
and so they're really good in the control of the getting having enough of the bacteria, but not too much.
Okay. And then Peter Cactus wants to know, where does the pollution go after the mushroom? Poof, it's gone?
No, because the fungus is taking the different parts of that chemical apart and redirecting it into not toxic materials. So that whatever that sequence of nitrogen, phosphorus, sodium, potassium, all of those things, they're all chewed up and reconsigned.
So really that's the probably the same answer for this question. Hemp removes heavy metals from the soil. Where does the hemp go? Is it being broken down?
Yep, in this case, it's being broken down so that the uh, the carbon in the hemp will be put into the structure of the body of the fungus. Some of it will still be in a form where the toxic material has been pulled apart as well and restructured so that it's not toxic.
The biology just loves all those compounds and uses it for something else.
That's right. Yeah. Yeah.
Nature.
Yeah, exactly. So, Peter Cactus again, are chop and drop plants placed around your crop a good idea?
Yeah, definitely. If you notice anything funky happening to those organic materials laying on the soil surface, just get out and put a really good compost extract on them. And
Mhm.
you'll shut down the diseases and the pest organisms. And then hopefully that gets all of the decomposition moving and going so that pretty soon you don't perceive that you're walking across those dead leaves of that dead plant material because now it looks like soil
Mm-hmm. Well,
because
I
it is.
know Whitcomb, the last show I was on with her, was mentioning not using the green, letting it actually dry first, and then putting it around the plant. She was worried about biology in the green material getting into the structure of the new plant. Would that be a problem?
I don't know why um, the green materials would be bad for a growing plant. The only time, if it was anaerobic, if it was not adequately aerated, like it matted down and,
Mm-hmm.
3 or 4 layers of leaves, one on top of the other, you'll get some anaerobic stuff going there. And so that might be the problem. So, she might want to, send me a little clearer description of what she was asking about so that I could answer the question for her.
Okay. I'll have to ask her on this Thursday's show. Perpetual has the last question, and it's more of a statement. What was monitoring for 18 hours in the pivot? It was the people that were monitoring and taking the samples, correct?
Right. Correct.
And that's it for questions from the audience. I hope I didn't miss anybody's questions, guys, but if I did, I'll apologize now.
I think there was one misnomer on the hemp. If the person is consuming the hemp, they do have to be careful because hemp's a dynamic accumulator and it will uptake some of the heavy metals. So that, yes, it's going to break down a lot of shit, but just like failed cannabis tests for heavy metals, it's one of those plants like a comfrey that'll pull all kinds of stuff up into the plant. So then the question is, well, when that plant dies, what do I do? Because now it's got accumulations. Well, it has broken a lot of it down, and yes, it's accumulated some of it. So just compost the plant, and it'll be gone the next, or hopefully, depending on how toxic your soil is. But it'll just keep breaking down and down and down. And I'm sure Elaine can go out on speak on that one.
Well, I was not thinking of the growing plant as part of that answer, because hemp to me is, it's like the strings corded, and that's what I was thinking of. Oops, there's another part to this question. Yeah, so you're right. Yeah, you've got to bear some care when you're harvesting and using soil that might have had contaminants in it.
Mm-hmm. Especially when you're using a dynamic accumulator as a plant to get that stuff out. What are you going to do with it? And the composting and taking it up to the thermophilic, etc., is part of the process.
Yep. If you do that, it's gone.
Yay!
And guess what, Elaine? You get to go home early if you want to. we got 3 minutes and 40 seconds. I don't know, Ab, did you want to add anything or ask her anything else?
No, just wanted to give my thanks and gratitude for taking time out on her birthday, but also for all the work that you've done and just getting a lot more women into science, getting a lot more women into agriculture, and then getting so many more citizen scientists and scientists excited about biology. So grateful for that work.
Well, I have to say thank you to all three of you, and the guys behind the scenes as well. So it's thank you for the chance to spread the knowledge about what's going on in the soil.
Well, and that's why we thank you, Doctor. Like, I know personally, I've been following you for quite a long time, and it's an amazing amount of knowledge that you have given to so many people globally, and is so greatly appreciated.
Thank you. Thank you very much. So we'll have to do this again sometime.
Yes, please, Elaine. Yes, please.
Oh, tease me. Yes, that's bad.
I will suffer my fate.
And we will let you go, young ladies, so that you can go and enjoy the roses that I hope your husband gave you on your birthday, and some cake that isn't going to be too much high in sugar.
Yeah, I think there's no hope there.
Well, Elaine, please do give Russ a big hug from me. And, it's always a pleasure to see you. And again, thank you so much for all the hard work that you do for humanity.
Well, thanks all of you who are doing this work too. So we shall continue on together.
There you go.
And with that, guys, I'm going to end the broadcast. We'll see you in a few days with Andy Lopez, the Invisible Guardian.